What is up, guys? Good Guy here. Today we're going to be talking about the endocannabinoid system. So what is the endocannabinoid system? Well, the endocannabinoid system is a collection of ligands and receptors throughout our body. The two known receptors for the endocannabinoid system are the CB1 and CB2 receptors. Both these receptors are G protein coupled receptors, and the CB1 receptor is located mainly in the brain but also operates in peripheral tissue. The CB2 receptors are located in the central nervous system, the peripheral nervous system, the immune system, the gut, and in white blood cells. Ligands in the endocannabinoid system are called cannabinoids. Cannabinoids that occur naturally in our body are called endocannabinoids. The two main endocannabinoids are anandamide and 2-AG. Anandamide will bind to CB1 receptors, but it can also bind to CB2 receptors. 2-AG is a full inhibitor of both receptors. Plant-derived cannabinoids are called phytocannabinoids. Our body will actually recognize phytocannabinoids as any other endocannabinoids. The two main phytocannabinoids, the two most widely recognized phytocannabinoids, are THC and CBD. THC and anandamide are actually very similar. The only main difference is that THC lasts longer and it's a lot more. It creates a stronger signal, if you will. Now let's talk about the efficiency of the endocannabinoid system. The endocannabinoid system is everywhere in your body. Wherever you point, you're either going to have a receptor or a couple cannabinoids floating through the blood. Cannabinoids are actually our body is actually able to synthesize and degrade cannabinoids very quickly. On the charts, you can see the anandamide is degraded by fatty acid amide hydrolase, and 2-AG is degraded by monocyclglycerol lipase. Both of these reactions produce arachidonic acid and two different byproducts. Now, because of the abundance of receptors in our body and the body's ability to synthesize and degrade cannabinoids so quickly, the endocannabinoid system is able to regulate with a lot of efficiency things like mitochondrial function, metabolic function, pain perception, memory, and apoptosis. Now let's talk about how the endocannabinoid system regulates mental function. Well, the endocannabinoid system can't just tell our brain to be happy or sad or to be sleepy or whatever. It has to manipulate this thing called GABA. GABA, or gamma aminobutyric acid, is a chief neuron inhibitor in the body. It suppresses neuron activity and dampens signals sent by neurons. On the right, you can see GABA. It is also a G protein coupled receptor, and it consists of 2 alpha subunits, 2 beta subunits, and a gamma subunit. Now, what the endocannabinoid system does is that when CB1 or CB2 receptors are activated, they reduce GABA expression, which will then reduce pain, increase your appetite, make you sleepier, reduce your anxiety. And when 2-AG is activated, it actually promotes GABA expression because it is an inhibitor of both receptors. Now let's talk about metabolic regulation. Well, the endocannabinoid system is able to regulate our metabolism through two main ways, actually. The most obvious way is through controlling our appetite. On the left, I have grossly oversimplified the hypothalamic reward pathway, which is a pathway associated with appetite. So basically what happens normally is that neurons send signals to other neurons telling the brain that, hey, we're hungry, or the body's hungry, I should say. And what GABA does, it actually suppresses the signals that neurons send to each other, or sometimes just blocks them, and it makes the intensity of our hunger way less intense. When the CB1 receptor is expressed, it suppresses GABA, and the intensity of signals between neurons is much stronger, which then increases our appetite. The second and lesser known way that the endocannabinoid system is able to regulate our metabolism is by the regulation of futile cycles. So futile metabolic cycles occur naturally in our body, like all the time. They don't really do anything, though. All they do is expend energy. And when the CB1 receptor is expressed, it prevents these futile cycles from occurring, which will then save energy that was being spent by the futile cycle. Through both these processes, the expression of the CB1 receptor promotes lipogenesis. But by these two same factors, again, chronic intake of phytocannabinoids like THC puts the user at risk of hepatic steatosis, which is the buildup of fatty cells in the liver. Now let's talk about mitochondrial regulation. The endocannabinoid system is able to regulate the mitochondria through a number of different ways. The main way is through like a bunch of different pathways. The main ones are calcium-based pathways. Also, other pathways that are like, there's so many of them. I'm only covering 3. So one of them on the left is by increasing the reactive oxidation species. So when CB1 receptors are expressed, they increase the reactive oxidation species, which will then induce an inflammatory cascade, which then induces mitochondrial stress, which will then increase our appetite because our mitochondria are working harder and they need more energy to support themselves. And now let's talk about the other way that the endocannabinoid system is able to regulate mitochondria. So basically, this other way is by the concentration of cannabinoids. The concentration of cannabinoids is directly linked to the mitochondrial health. A low concentration is good for mitochondria, and a high concentration is bad. Due to this, all mitochondria are equipped with a fatty acid amide hydrolase, which will degrade the cannabinoids if the concentration becomes too high. Now let's talk about the apoptotic regulation of the endocannabinoid system. In a study done on lab rats, THC was found to induce apoptosis. The endocannabinoid system does this through the ceramide ERK signaling cascade. So basically what this is that when CB1 or CB2 is expressed, it starts this process called ceramide synthesis, which will then of course produce ceramide and start another process called the ERK signaling cascade. What the signaling cascade results in is the cellular arrest and apoptosis of the cell. Now that we have a basic understanding of the endocannabinoid system and some functions that it regulates, let's talk about some possible treatments derived from the manipulation of the endocannabinoid system. So the manipulation of the endocannabinoid system is hypothesized and proven to be potential treatments for ALS, autism, epilepsy, HIV, depression, anxiety, and cancer. The manipulation of the endocannabinoid system is especially efficient for cancer because not only does it increase the calorie intake and reduce the energy expenditure, but it also promotes apoptosis, which is the main problem with cancer, is that your cells are they're not going through apoptosis correctly. But I digress. I'm not going to go into cancer. The 2 main ones that we're going to be focusing on today are anxiety and depression. So let's look at the 2 main treatments for anxiety and depression. For depression, we have selective serotonin reuptake inhibitors, and for anxiety, we also have selective serotonin reuptake inhibitors, but one treatment that I'm going to focus on today are benzos. So let's look at a healthy serotonin synapse. So on the left, you can see a healthy serotonin synapse, and you can see vesicles of serotonin being transported into the synapse from the presynaptic nerve ending. Now the postsynaptic nerve ending will bind to the serotonin from the synapse, and once enough serotonin have bound to receptors on the postsynaptic nerve ending, it will send a signal to the next nerve, starting this process all over again. Now you can also see another opening to the right on the right side of this presynaptic nerve ending, which is actually an opening to reabsorb serotonin from the synapse. Now the problem is that in depressed people, they don't have enough serotonin in the synapse to send a signal. So basically, this just leaves them feeling like crap because they don't have enough neuron activity, and they're just feeling like, oh my god, So a solution that that scientists have came up with is the selective serotonin reuptake inhibitors. Basically what these do is they block the reabsorption of serotonin from the synapse, and this will then amplify the signal of serotonin, or amplify the concentration of serotonin in the synapse, which will then amplify the signal. This is because the presynaptic nerve ending isn't reabsorbing serotonin anymore. Now let's look at the treatment for anxiety. So as I've mentioned before, GABA is in control of a number of different systems. One of them is including anxiety. What happens when GABA is overexpressed is that anxiety is a result of that. So what benzos do is they actually bind to the alpha and gamma pocket, and they cause a conformational change in the structure of GABA, which will render it useless for the time that benzos are attached to it. Now let's look at the problems of both these treatments. They seem perfectly fine, right? Well, actually, the SSRIs are kind of inefficient because they only treat specific neurotransmitters. So it's kind of like a temporary treatment, and a lot of times problems come back even worse, and like with worse thoughts of depression and suicidal thoughts. Oh my god, I messed that up. But none of the matter. And the problem with benzos is that they're extremely addictive, because GABA is mainly in the limbic pathway, or not mainly. It's heavily associated in the limbic pathway, which is the pathway most closely associated with addiction. Another huge factor to addiction in benzos is the extremely short half-life of them, which will just have that results in the user having to take more and more and more, or just to like get treated, which this is why it's like extremely addictive. And somehow it's a Schedule 4 drug, which means that it's not addictive. It definitely is addictive. But the current endocannabinoid system treatments aren't really that efficient either. They mainly consist of flooding our bodies with phytocannabinoids like THC and CBD. For some diseases, this is actually very efficient, like for epilepsy and cancer. The flooding your body with THC is actually a pretty dang good strategy. But for other illnesses like anxiety, depression, ALS, autism, HIV, we have to be a little bit more precise in the way that we treat it. Just flooding our bodies with phytocannabinoids isn't going to work. Scientists have hypothesized like we could like selectively inhibit some factors of the endocannabinoid system, like we could inhibit fatty acid amide hydrolase to increase the concentration of anandamide, or we could inhibit anandamide producers so that 2-AG is the main cannabinoid present in our body, which will then increase GABA production, which can be bad if you don't have, or GABA expression, as to say, because like sometimes you don't have enough GABA and you just your brain is just overactive. So right now we don't have the resource, or scientists don't have the resources disposable to them to be able to develop these advanced treatments. And this is just a shame, because the endocannabinoid system is so integrated into our body that not studying it as much as we have is just like, it's so understudied. The only reason it is so understudied is because it is a Schedule 1 drug, or the marijuana is. I don't know, I'm getting off on a tangent here. But basically, that is my whole presentation. I hope you guys enjoyed. And like and subscribe and comment and like and all that crap, share with your friends. Tell them, oh my god, this is the best video ever. We're going to come back at it with another video. Next video I'm going to teach you guys how to 360. Alright, peace.